I have do this lecture like 6 times. This is the second part of the lecture 6. And based on so every lecture I still make some modifications to improve it. So from this lecture I will start to add some first one I want to add a reference list at the last page. And if there's something happened in the world, in this society, maybe I will combine them together. So today is a new try. So for me, it's try something new that I will try to talk about some news then related with some basic scientific knowledge that will not be changed and will not be forgotten forever. Like today, I'm going to, because recently we know that the Egypt Suez River, the canal, Suez Canal, is being blocked by a super large ship. And so, and when I'm reading this chapter, the chapter 6, it happens there's a rhodopsin functional model. It's named as a Panama Canal model. So I think that's very super interesting. And then the second part is a model that use the software and use machinery and deep learning to analyze the unknown rhodopsin structures. And this make me remember about when I find those text documents. So we all know that those text forms are super hard to fill in. But after so before, like I think I made some mistakes and I mail in and I get the mail back from IRS. And in recent years, I started to use the software HR Block. And when using this software, I feel it's quite similar with this ARM model that we're going to talking about today. Is that it will ask you a lot of questions, and it also have several like a default setting that if you don't know, then you just use a default setting. I think that's pretty cool, and that's very easier to use, and I believe it will give a correct, like precise and clear result, no matter it's a tax return or a rhodopsin modeling. Let me see this. Yes. Okay. Now, yeah, I can hear that. Okay. Why every time I have to talk about something that's not directly with the lecture that I really, because there's a delay between the Zoom software and and YouTube, and so so I prefer that I don't want like repeat it again. So I just say something that before I can hear, and now I can hear that. Then I will start now. I'm going to share my screen, and this is today's also the lecture 6 into the chapter 6 and of the part 1. Oh, I'm sorry. It's I modified sometimes part 1. Yes. The part 1 lecture and the model of chain rhodopsin. And we're going to talking about the rhodopsin, like it's a functional model. It's called Panama Canal model. And the second part is our R model. That's what I mentioned that it can close to the tax return software. This Panama Canal model, so we know that just a few days ago, the Egypt Suez Canal is getting stuck by a ship. And so all these riverways is being blocked. And during I give this lecture, I think probably like the workers in Egypt are still working very hard to reopen this canal. And in this world, there are several very famous canal, and another one is that link to the North American and South American. That's the Panama Canal. Panama Canal is located at Costa Rica, I think, between Panama and for this canal, there's a very a specific structure is here, because between these two sides the water level were different, and for ship, those that were passed from like the lower water surface to go to a higher part, then they need this canal, this artificial structure of this canal. That is, there's some gates. There are two gates. So the ships first go in the first gate open. The ship drive into the center part. Then the first gate closed and raise the door and put the water into the center part, and the water level is being increased and until it's at the same level at the higher part of the water surface. Then the second gate is open and the ship can go to the higher water surface. So this is how this Panama Canal works. So when I see this model, I think it's quite clever, because for the pump's function, for the pump, it's always like take some energy that pump the iron or cation from the low concentration area, then go to a higher concentration area. That's how the pump work, and pump require energy. So for the Panama Canal, then maybe like there's some electricity energy and some mechanical energy that they need to put the water into the center part. And for the proton pump, is the energy is coming from a photon. And this photon and the whole structure of rhodopsin is that they can convert this energy from the photon, capture the photon, then cause a conformation change, and then make these pumps opening. Like for this state, it's like the pumping out of some stuff. Like the default setting is the intracellular part is open, then conformation change to switch to, and the open gate is the outside gate is open. So for each pump, it's like it has two doors. So the default at the resting state, the inner door is open, and after there's a conformation change, the inner door is closed and the outer door is open. So this I think from the energy side of view, we can confirm that, we can say that it's really can be comparable to the canal. So we just mentioned in last lecture that many structure studies suggest that the structure of bacterial rhodopsin switch from the outward open to inward open state is during this M1 to M2 status. So auto capture the photon is at the resting state to the K status, but this energy conversion is at the M1 and the M2 status. At this status, the outward opening will change to inward opening state. And this, so here I just give a very very generous like instruction of the Panama canals. And yes, for the rhodopsin structure, there are several parts that's very important for its function, like the ion selective filter, like the Schiffer base, the Schiffer base, protonized base linked with the lysis residue, and this is like the gate or the lock, and an exit gate and extracellular the sodium binding site. For majority of those rhodopsins, we can tell the extracellular part and the cytoplasma part is because at the extracellular part there's a beta sheet folding at the extracellular part. Yeah, because scientists sometimes people prefer like put the extracellular part on the top, but someone prefer to put the cytoplasma part on the top. Either way is in published data, but I think scientists will know that because there's a beta sheet always at the extracellular part. So okay. Yeah, I think I already finished like all the details that we need to. So in this article, actually I think it's a review that published 2 years ago. This Panama Canal model, I think it's a very um enlightful idea to understand the pumps function. Yes. So let's move on to a second part of today's lecture is that, and here I'm going to introduce an automatic rhodopsin model. This automatic rhodopsin model is it's kind of like a software that can it's based on some already known X-ray crystallization experiment data. The structure analyzed based on like 35 structure. Then they try to align their calculated model to the experimental model, experiment data, the facts. And after quite align with these two together, then this model will be a quite like precisely model that can simulate those different rhodopsin, known or unknown. So there's several very critical parts of rhodopsin. That one is the retinal chromophore cavity, and also need to find that those protonized status of this ionizable residue, like those residues, some residues are aligned within those channels or the pumps pathway in the pathway of the cation or the ions, and those residues that if it's ionizable, it need to be highlighted. And also some residues that in the extracellular part or intracellular part, and because they can like function as an ion selective filter, and that's also important for the structure or the function of the rhodopsin. And also need to find that the counter ions as for like when we talk about channel, it's like the channels are open and it's open. But for pump, the the counter ions are very important, because we remember that every the protonized site, the proton will jump in from one side to another side to another side to another side. That those every side of these counter ions will be very critical for the function of the photocurrent. So the most important part and the meaning of a software model is that it can predict the result of a single or multiple mutations result. That's the last part, that the final congruous generation of single or multiple mutations. And this alpha-arm model system is based on the QM and MM model as a quantum mechanical, molecular mechanical model. What's important is that because the protonized the Schiffer base part is very important and it's very critical. So its surroundings determine the central function of this rhodopsin. So this part need to be focused at the quantum mechanical level to get a meaningful result. But for the outside, for the backbone structure of the transmembrane, maybe just require an extensive and time-saving molecular mechanical model. So I need to combine these two. So this makes me think about like the human eyes. We all know that the human eyes have a fovea, and there's a lot of cone opsin, that concentration over there, and outside the cone for photoreceptor is quite sparse, and that rod photoreceptor over there that. So this is design is quite similar to like the retina anatomical structure, that something need to be focused and something need to give a background, a general picture. So that's the QM and M model. And this study, the arm, is the best on this QM and MM model, but it improve it and have some other advantages. So what's the QM and MM model disadvantage? First, it's time consuming. And the second, because every data, every like index need to be input by the people. So different scientists they put in some what they think that's the most important part, but maybe different from another scientist. So then the final result will be no compatible. So because there's some human error, error like factors exist, and there's also depending on like in the scientist is experienced or unexperienced. So this model is an error prone. So basically, so this arm model is, of course, it's a basic idea. It's quite similar to the QM and M model, that it need to input like an X-ray crystallographic structure and the PDB bank protein model information, and also need to put the list of residue from the chromophore cavity, and need to input the counter ion information, and outside and also outside the ionizable side chains, everything need to be input. But the advantage of the R model is that it has a default choice and answer several, and the scientist can answer some very specific questions, then to use it as an input for this model. So it's like this model will ask you some questions, then specifically to align these models' results. So generally say that this model have the first step, that the initial setup of the working the PDB file, and the second step, it will automatic generation of the cavity, and it will give out analyze, it will give out the ionizable state and called the ion placement. So the whole flow chart is shown like this. And the let me see, it's not here. Oh, okay. So for this model, they also analyze the PUTION code, the Prussian code. And this code is also analyzed that it provides an intracellular part. This it is positive charged or is a negative charged. So you calculate all the positive, negative, and the total for the intracellular part is positive charged, and the total for the extracellular part is negative charged. It's a negative charged, so it can attract like the cations, like sodium, and in the intracellular part, it's positive charged, and it were covered with a negative charged ions like chloride. So we all know that for most of those cells, that the intracellular part is a negative charged. It's more negative comparing to the outside. So this is the cool part of this model, because it has a function that it has a built-in mutation module that it can automatically generation of mutant by using this software. That it can generate a mutant and mutant another amino acid, and so this is all can be done on the software. So and there's no need for those tedious work for like a single point mutation, then get the construct, and those construct then go to transfection, then go to recordings and everything. And this will save a lot of time and maybe more precisely. It's because like for like a transfection, we know that the dosage of the DNA, the status of the cell, and never mind like the person who do the recording, do the transfection for all this, these are all the variant. But for this model, those variant will just not exist because it's on based on those equations and previous data structure and give this prediction. That's cool. That's super cool. So here it shows some results that the result that they are like give out some examples like BPR like this, and the rhodopsin and BRAT, and the core part as a given mutation that is modified. It's the same. It's based on the same like ancient it cannot be said ancient, like there's the same molecule but the given mutation, and see the mutation part is the critical residue is still the same, but the mutation is in another areas. So that this is the most cool part of this model. So for this model, I can give out several important informations like the main chains. They give a sign carton the background, and it will give out the chromophone, the protonized Schiffer base, and the cavity, and the main counter ion. The four is here. Oh, the three is the lysis. counter ion, and the secondary, also secondary counter ion. I see another color approached here, and protonized residue, and external chloride, and maybe internal also water molecules such as tubulin. And we remember that water molecules inside this cavity will also be very important to form those hydrogen bond networks. And the residue of the chromophore and these cavities subsystems. And this part is the QM, the Kuhlmann level, that the central focus part. Yeah, this is how this system goes, and this is a reference. So we're talking about is based on these studies. And thank you.